So picture this. You've just finished a massive, incredibly heavy meal.
Oh, yeah. We all know that feeling.
Right. Let's say it's exactly the kind of meal that's actually drawn in the graphical abstract of the research we're diving into today.
Right, the burger.
Yeah, a massive double patty burger, just absolutely dripping with cheese, and maybe, I don't know, a huge slice of pepperoni pizza on the side.
It's just a totally casual Tuesday lunch.
Exactly. And you know that immediate, just heavy sluggishness that hits you maybe an hour later?
The food coma.
Yes, the classic food coma. We usually just rub our bellies and laugh it off, right? Like, oh, I eat too much.
Yeah, time for a nap.
But what if that sluggish feeling is actually the echo of a rapid hidden war being waged inside your gut?
It's a crazy thought.
It really is. So welcome to the deep dive. Today, we're unpacking a really groundbreaking paper published in the journal Immunity. And our mission here is to figure out a truly startling discovery.
Which is
how a high-fat diet causes immediate violent chaos in your intestinal immune system, starting long, long before a single pound of weight is ever gained.
Yeah, and it really is a profound shift in how we understand the relationship between, nutrition and immunity.
Right, because normally we think of that as a slow process.
Exactly. Traditionally, when we talk about diet-related immune issues or metabolic diseases, we're looking at a timeline of years or even decades.
Like getting diabetes in your 50s after a lifetime of bad eating.
Right. The baseline assumption in the medical community has almost always been that immune dysfunction is, well, it's a secondary consequence.
No, I see.
We think of it as the result of chronic, long-term conditions, like carrying extra weight for years, developing insulin resistance, metabolic syndrome.
The slow wear and tear.
Exactly. The slow, gradual wear and tear of a typical Western lifestyle.
Okay, well, let's unpack this, because here is the massive aha moment from this stack of research.
Yeah, this blew my mind.
The scientists running this study found that the damage to your gut immunity, it doesn't need months or years to develop. It actually happens completely independently of caloric excess.
Which is wild.
It happens before there is any measurable weight gain, way before there is any glucose intolerance. We're talking about severe immune impairment that manifests within hours, or, like, at most, days of consuming a high-fat diet.
Which is incredibly fast for an immune shift of this magnitude.
Yeah.
I mean, to understand how something so rapid could occur, we really have to look at the very first domino to fall in this whole biological cascade.
Okay, and what is that first domino?
Well, it brings us directly to the physical barrier of your gut and the trillions of microbes living right outside of it.
Right, the initial breach. I mean, if we want to understand this rapid change, we have to look at the border wall itself. So what exactly is happening down there in the dark right after we finish that double cheeseburger?
So within a very short period of high-fat diet intake, your body experiences a state called dysbiosis.
Dysbiosis. Let's break that down for everyone.
Yeah, dysbiosis is essentially an abrupt, chaotic imbalance in the bacterial populations of your gut.
Like the good guys and bad guys get totally out of whack.
Exactly. You have to realize the high concentration of dietary fats you just ate, it acts as a massive environmental shock down there.
Oh, wow.
It's like sudden climate change for your microbiome. It rapidly alters which microbes thrive and which starve.
That makes sense.
And this sudden bacterial shift has immediate structural consequences for your gut wall. It causes a rapid reduction in the production of intestinal mucus.
The protective slime, basically.
Yeah, the slime.
Yeah.
And it physically compromises what we call the tight junctions of the intestinal epithelial barrier.
Okay, tight junctions. I think I'm visualizing this. Let me try an analogy here.
Go for it.
It sounds almost like a medieval siege.
Yep.
Like if we imagine your gut lining as a giant medieval castle wall that's protecting your internal bloodstream, then that mucus layer you mentioned is the deep protective moat on the outside. And those tight junctions, which are literally just protein complexes stitching your intestinal cells together, those are the heavy wooden gates.
That's a perfect way to picture it.
So this sudden influx of a high-fat diet acts like an instant severe drought that dries up the moat while simultaneously, I don't know, wresting the hinges right off those gates.
It captures the structural failure perfectly. Because when that moat dries up and the gates lose their integrity, your gut barrier becomes highly permeable.
A leaky gut.
Exactly. The immediate threat is that microscopic invaders, specifically microbial products like lipopolysaccharides or LPS.
LPS, okay.
Yeah, which are basically just fragments from the cell walls of bacteria. These LPS fragments slip through those broken gates and enter your sensitive intestinal tissue.
Oh, that sounds bad.
It is. And once these microbial products cross the barrier, they immediately encounter your castle guards.
Ah, the immune system steps in.
Right. In cellular terms, we're talking about mononuclear phagocytes or MNPs.
MNPs. These are basically the sentinels pacing the courtyards, right? Like the macrophages and dendritic cells that just patrol our tissues looking for trouble.
That's exactly their role. They are the body's localized alarm system.
Okay.
When MNPs detect these invading bacterial fragments that absolutely shouldn't be there, they become highly activated.
Like they wake up.
They transition instantly from a state of quiet, sleepy surveillance into a state of aggressive defense. They start releasing a massive flood of inflammatory signals.
Like what kind of signals?
Specific chemicals known as cytokines. They shoot these straight into the surrounding intestinal tissue to sound the alarm.
Okay, so let's do a quick recap here for everyone listening. If I'm tracking with you so far, you eat a high-fat meal, it immediately shocks your gut bacteria into an imbalance.
Right, the dysbiosis.
That imbalance dries up your protective mucus moat and breaks open the gates of your intestinal wall.
The tight junctions fail.
Bacterial junk slips through and your MNP guards start screaming and throwing inflammatory alarms everywhere.
You've got it.
So I want you to ask yourself, you listening right now, next time you eat a heavily processed high-fat meal, are you thinking about the immediate structural changes happening to your inner castle walls?
Probably not.
Right, most of us aren't. But an alarm usually means innocent bystanders get caught in the chaos. Who exactly is caught in the crossfire of this massive inflammatory alarm?
Well, the primary victims of this localized inflammation are a highly specialized group of cells called Group 3 innate lymphoid cells, or ILC3s.
ILC3s. Okay, this is the main character of our paper today.
Right.
What exactly makes these cells so important? Like, what do they do for us?
So to really understand their importance, you have to separate them from the immune cells most people know about.
Like white blood cells, T cells.
Yeah, exactly. They're not like your standard adaptive immune cells like T cells, which can take days or weeks to build an army and mount a specific response to a specific virus.
Oh, okay.
ILC3s are part of the innate immune system. They are the crucial rapid response sensors of the intestinal environment.
Like the beat cops.
Yes. Think of them as the local peacekeepers. Their entire job is to sit right at that mucosal border and integrate signals from the food you eat and the microbes that live there.
They sound super busy.
They are. They're constantly releasing molecules that help maintain the integrity of those castle gates while also telling the immune system to tolerate friendly bacteria and simultaneously warding off actual bad pathogens.
Wow.
They are, without exaggeration, the ultimate maintainers of intestinal homeostasis.
They sound incredibly vital. So what happens to these vital peacekeepers when those MNP sentinels sound the massive inflammatory alarm we just talked about?
The research paper reveals something quite staggering. In both mice that were fed a short-term high-fat diet and in intestinal biopsies taken from human individuals who are overweight or obese,
okay, yeah,
the populations of these intestinal ILC3s are drastically reduced. They essentially vanish from the tissue.
Wait, let me push back on that for a second. When you say they vanish, are they just migrating somewhere else in the body to hide out until the inflammation dies down? Or are they actually dying?
That is a critical distinction, and the researchers looked really closely at this. They are not hiding. They are not migrating to the lymph nodes to wait it out. They're dying.
Oh, wow.
And the specific manner in which they die is highly violent and mechanically unique.
What do you mean by mechanically unique?
Well, it is not a standard, quiet, programmed cell death where the cell just politely recycles itself. It is a total metabolic catastrophe triggered by the bizarre combination of the fat they are absorbing from the diet and the inflammation they are experiencing from the guards.
Okay, let's make sure we've really got this locked in before we go further.
Good idea.
These ILC3s are the essential rapid response peacekeepers of your gut.
Yeah.
But when exposed to this immediate high-fat diet inflammation, they don't just clock out for the day, they violently die off. They rapidly disappear from your intestine.
Right.
So to everyone listening, think about how your city maintains peace. If the first responders are completely wiped out on day one of a new crisis, what happens to your body's ability to handle everyday normal threats?
It plummets.
Exactly. Let's zoom in on that violent death, though, because this is where the biology gets wild. How exactly does a combination of fat and inflammation physically kill a cell?
What's fascinating here is the precise cellular mechanics of this death. The researchers mapped it out completely, and it all centers around the ILC3's mitochondria.
The powerhouse of the cell. We all remember that from high school biology.
Exactly, the power plants. Now, under normal, healthy conditions, ILC3s are actually quite good at handling dietary fats.
Really? I would have thought fat was just bad for them.
No. They take up lipids from your food, and they burn them for energy to do their peacekeeping jobs.
Oh, that makes sense.
They do this using a highly specific biochemical pathway called PPAR alpha-dependent fatty acid oxidation, or FAO for short.
PPAR alpha.
Yeah, you can think of PPAR alpha as a molecular switch.
Yep.
It turns on the necessary genes required to burn fat safely inside the mitochondria.
So normally, fat is just fuel. They absorb it, flip the switch, burn it, no problem.
Correct. But here is the fatal disruption.
Okay, brace yourselves.
When those MMP guards release those inflammatory panic signals, specifically a cytokine called TNF-alpha,
right.
those signals act directly on the ILC3s and actively suppress that PPAR alpha-dependent pathway.
Wait, they shut off the switch?
Exactly. They turn off the switch. And interestingly, the researchers noted this suppression is highly specific to our peacekeeper ILC3s. It does not happen in other immune cells that actually promote inflammation, like Th17 cells.
Wow, so the peacekeepers are uniquely vulnerable.
Very much so. So the ILC3 is still sitting there absorbing massive amounts of fat from the burger you just ate, but the inflammation has suddenly turned off its ability to burn that fat.
Oh, wow. Okay, I think I have another analogy for this one.
Let's hear it.
Imagine the ILC3 cell's mitochondria as a heavy-duty wood-burning stove inside a house.
Okay, I'm with you.
Under normal conditions, you put logs in, they burn safely, you get heat to power the house. Eating this high-fat diet is like someone shoveling an endless supply of logs, which are the lipids, into that stove nonstop.
Right, way too much fuel.
But the sudden inflammation from the MNPs acts like someone aggressively throwing a wet blanket over the chimney, totally suppressing the ability to burn the wood.
Yes.
The wood just piles up, it can't burn properly, the room instantly fills with thick, toxic smoke, and the house eventually burns down from the inside.
That is a phenomenal way to illustrate the concept of lipotoxicity.
Lipotoxicity.
Yeah, because that toxic smoke in your wood-burning stove analogy represents exactly what biochemists call mitochondrial lipid peroxidation.
That sounds intense.
It is, because the fats can't be safely oxidized for energy, they just accumulate inside the cell and become highly reactive, unstable lipid peroxides.
A toxic smoke.
Exactly. These toxic molecules are like biological shrapnel. They aggressively attack the membranes of the mitochondria.
Oh, man.
The mitochondria lose their structural integrity, they literally rupture, and this catastrophic internal damage causes the rapid, violent death of the entire ILC3 cell.
Now, hold on, because I read a fair amount about longevity and the various ways cells die. And usually when we hear about lipid peroxidation causing a cell to die, it's a very trendy, specific pathway called ferroptosis.
Right, ferroptosis.
Which is an iron-dependent cell death. Is that what we are looking at here? Like, is the high-fat diet causing ferroptosis in our gut?
It's a very logical assumption. And frankly, it is exactly what the researchers initially hypothesized when they saw all those lipid peroxides.
Oh, really?
Yeah, but they explicitly tested for it, and the answer is a definitive no.
Wait, really? Not ferroptosis?
Not ferroptosis. They looked at the specific transcriptional signatures, meaning they checked to see if the specific genes that govern ferroptosis were turned on or off.
Okay.
They checked both in living organisms and in lab cultures. The ILC3 cell death occurred completely independently of any ferroptosis pathways.
Huh.
Furthermore, it wasn't rescued by antioxidant interventions that usually stop ferroptosis in its tracks.
Oh, wow. So what does that mean?
It proves we are looking at a highly unique, newly identified immunometabolic vulnerability that is specific just to these peacekeeper cells.
That is wild. They found a totally unique way for these cells to choke to death.
Yeah.
But it begs a massive question about the science itself. Like, how can researchers possibly know exactly what is happening inside the microscopic mitochondria of a tiny immune cell?
It sounds impossible, right?
Yeah. How do you physically prove the chimney is blocked and the smoke is building up in real time?
Well, the methodology they used here is truly elegant. It's a testament to modern science. They used live cell imaging with a highly advanced piece of equipment called a spinning disc confocal microscope.
Spinning disc confocal microscope. Okay, sounds expensive.
Oh, very. This isn't your high school microscope. It uses lasers to scan multiple points across the cell simultaneously, allowing them to watch living cells in real time in 3 dimensions without frying them.
That's incredible.
And to see the specific mechanism, they applied 3 specific fluorescent dyes.
Okay, walk us through the dyes.
First, a dye called lipotoxic. This literally lights up bright green only when it binds to those toxic lipid peroxides.
Okay, so green means toxic smoke.
Exactly. Second, Mitochondrial deep red. This glows deep red only when a mitochondrion is healthy and functionally burning fuel.
Red is good. Red is the healthy stove.
Right. And finally, propydium iodide. This is a dye that absolutely cannot enter a healthy cell, but it rushes in to stain the cell's DNA, bright red, the exact second a cell membrane ruptures and dies.
So they basically created a microscopic traffic light system.
Essentially, yes. By watching these colors shift under the laser microscope, they literally watch the deep red functional mitochondria fade out, then the bright green toxic lipid peroxides accumulating.
The green smoke taking over.
And then the sudden flash of the propydium iodide as the cells violently died.
Wow.
They watched the lipotoxicity happen live.
That is both incredible and slightly terrifying to picture happening in my own stomach right now.
Definitely.
So let's just make sure we have this mechanism straight. Another recap for you guys listening.
Yeah.
The core issue here is a literal metabolic chokehold. The inflammation from the gut barrier breaking down blocks the ILC3's ability to burn the fat it's actively absorbing. This causes highly toxic lipid peroxides to build up, which destroys the cell's power plants and kills the cell entirely.
Beautifully summarized.
So ask yourself, are you starting to see how the food you eat acts as immediate biochemical information, instructing your cells on how to behave, rather than just acting as a simple calorie count?
It really changes how you look at a menu.
It really does. But let's take a step back, because a lot of really cool scientific deep dives end with a frustrating caveat.
Oh, I know what you're going to say.
You read this amazing paper, and at the very end it says, well, this is fascinating, but it only happens in a sterile lab mouse.
The classic trap.
Right. So does this chokehold actually happen in us?
Well, if we connect this to the bigger picture, the answer is a definitive yes. This is not just a mouse phenomenon.
Oh, thank goodness. I mean, not thank goodness, but you know what I mean.
I do. The researchers knew they had to prove human relevance, so they analyzed human intestinal tissue, specifically ileal and jejunal biopsies.
Which are?
Sections taken straight from the human small intestine, from individuals clinically classified as obese.
Okay, real human tissue.
Right. And when the researchers isolated the human ILC3s from these biopsies, they observed the exact same molecular impairment.
Wow.
The human peacekeeper cells exhibited the very same dangerous suppression of fatty acid oxidation. They were choked out, just like the mice cells.
So it's totally the same.
The vulnerability is conserved entirely across species.
Well, here's where it gets really interesting, though. We spent this entire time talking about the damage, right?
The doom and gloom.
The drying moats, the dying cells, the blocked chimneys. It sounds pretty bleak. But this isn't a story of permanent doom. The researchers actually found a massive silver lining in all this destruction, right?
They did, yes.
Yeah.
And it centers on the incredible concept of reversibility.
Reversibility.
Despite how violent and rapid the cell death is, and how quickly the gut is depleted of its peacekeepers, the overall situation is highly malleable.
Right. Because if you look at the graphical abstract of the paper, which is basically a comic strip summary of the research.
Yeah, the diagrams are great.
On one side, you have the double burger and the pepperoni pizza causing the damage and the red inflammation. But on the other side, there's a giant arrow pointing to a return to health, symbolized by watermelon, fresh greens, grapes, bananas.
Exactly.
So what exactly is the antidote here? How do we fix the broken stove?
The antidote is stunningly simple in its mechanism. It's dietary normalization.
Just eating better.
Yeah. The researchers found that they didn't need a complex pharmaceutical drug to fix this. By simply removing the excess heavy fats from the diet and returning the subjects to a standard balanced diet.
Represented by the greens and fruits.
Right. The intense selective pressure on the gut was lifted almost as quickly as it started.
Walk me through what happens physically during that reversal. Does the moat fill back up?
Exactly. When the high-fat shock is removed, the dysbiosis begins to resolve. The bacterial populations stabilize.
The good guys return.
Yep. Because the bacteria are stable, the intestinal cells can repair the tight junctions and produce mucus again.
The gates are fixed.
Once the gates are closed, the bacterial products stop leaking through. And without those leaks, the M&P guards realize the threat is over.
They stand down.
They quiet down, and they shut off the inflammatory alarm.
And without the alarm, the chimney unblocks.
Precisely. Without the TNF-alpha inflammation suppressing it, the PPAR alpha switch in the surviving ILC3s turns back on.
Wow.
The cells regain their ability to safely burn fat through oxidation. The toxic lipid peroxide smoke clears out, the lipid toxicity stops.
The green smoke is gone.
Gone. And the populations of these vital immune peacekeepers completely restore themselves in the intestinal tissue.
So if I'm understanding this completely, let me just wrap my head around this. This severe immune vulnerability, this metabolic chokehold, is absolutely present in humans. But, and this is a massive but, simply restoring a normal, healthy balance of macronutrients to your diet can rapidly rescue these vital immune cells and reverse the damage, sometimes within days.
It's incredible how resilient the body is.
Think about the agency that gives you, the listener. How quickly could you reset your own gut immunity, shore up your castle walls, and bring your peacekeepers back online just by changing the makeup of your next few meals?
It is a very empowering thought. It forces us to view diet not just as a long-term driver of weight or metabolic syndrome over decades, but as an immediate dial-like modulator of our frontline immune defenses.
Exactly. So what does this all mean for us walking around in the real world? Let's bring it all together.
Let's do it.
We started this deep dive looking at a plate of heavy, high-fat food. We now know that the consequences of that meal aren't waiting for you a decade down the line when you step on a scale or go to the doctor for a physical.
Right, it's happening now.
It is a rapid, hours-long mechanism. The fat abruptly shifts your microbiome. That shift breaks down the physical mucus moat and the cellular gates of your gut barrier. Microbes slip through, triggering your local guards to flood the area with panic and inflammation.
The sirens go off.
And that inflammation acts as a metabolic chokehold, literally suffocating your gut's most important immune peacekeepers by causing toxic smoke to build up in their power plants.
Yep.
But crucially, we also know that the very next meal you choose to eat has the power to start reversing that entire process.
The immediacy really is the key takeaway here. Your daily, even meal-to-meal dietary choices have an instantaneous and profound impact on the structural integrity and defensive capabilities of your entire immune system.
Absolutely.
And knowing that, I want to leave you with one final, perhaps slightly unsettling dynamic to consider.
Oh, I love a good lingering thought to chew on. What do you have for us?
Well, we now know with absolute certainty from this paper that a sudden high-fat meal rapidly depletes your ILC3 peacekeepers, leaving the gut physically and immunologically highly vulnerable.
Yeah.
Consider this scenario. What happens if you consume a heavily contaminated piece of food, say a dangerous pathogen like salmonella?
Oh, no.
At the exact same time, you eat a massive high-fat meal.
Wait.
If your immune first responders, the cells whose exact job is to fight off things like salmonella, are being actively wiped out by fat-induced lipotoxicity at that very second, does that high-fat meal essentially act as a Trojan horse?
Oh my God.
Does it leave the castle gates wide open for a severe systemic infection that your body would otherwise easily fight off on a normal day?
Wow.
It completely reframes how we think about not just what we eat, but the timing of what we eat and what dangerous microbes we might accidentally be eating it with.
Wow. A Trojan horse for pathogens. That is a brilliant and honestly terrifying connection. That is definitely going to make me look very differently at grabbing a greasy late-night burger from a questionable food truck.
Definitely a risky move.
Next time you feel that heavy sluggishness after a massive meal, remember, it might not just be a food coma. It might just be the smoke clearing from the battle inside your castle walls. Thank you so much for joining us on this deep dive. Keep questioning the science of your everyday life, pay attention to your peacekeepers, and we will catch you next time.